great pleasure. I get to introduce Kristen Gorman. Kristen's a new scientist at the Prince William Sound Science Center. And prior to her arrival, she did her PhD research down in Antarctica at Palmer Research Station on penguins, and has defended and all that, so she is now officially Dr. Kristen Gorman. And prior to that, she had done a lot of work up here in Alaska, did her master's research up here in Alaska, right?
Western Alaska, yeah.
In Western Alaska, on ducks in Scotland, right? So we're very pleased to have her here tonight to talk about penguins in Antarctica. So, Kristen?
Hey, thank you, Mary Ann. I apologize, I think the slides are a little bit dim with this light. So
it's not the slides, it's that the sun is bright.
Yeah.
Have sun?
So as Mary Ann mentioned, what I'm going to talk about tonight is really all the work I did, which was part of my PhD thesis at Simon Fraser University. And by coincidence, the producer and director of People of a Feather that we're going to be showing tomorrow night also did his PhD at Simon Fraser University. So it's an excellent movie, and I think you'd really enjoy it. Yeah, as I said, I'm going to talk about my penguin work. And I just want to mention my two advisors, Tony Williams, who's at Simon Fraser University, and also Bill Fraser, who works with the Palmer Station Antarctica Long-Term Ecological Research Program. So this is an LTER site that's funded by the National Science Foundation. And Bill has worked in Antarctica for his entire life. He did his PhD there, and they've both been incredible mentors, and I wouldn't have had all these great experiences without them. So I'm going to focus on the three species of penguins that I worked on. This is the Adelie penguin here on the left, and chinstrap penguins in the middle, and then gentoo penguins on the right. And really what we're going to talk about today is aspects of environmental change and penguin response to environmental change, and what the mechanisms might be that are linking the two. Just to give you guys a bit of orientation, here's Antarctica, down in the southern hemisphere, and so we work here on the Antarctic Peninsula. And so we're just located just south of Patagonia, and then you can't really see, but this is Africa here, and then New Zealand and Australia. So the three U.S. research stations in Antarctica are Palmer, South Pole, and McMurdo. So we access Palmer Station through South America, but if you go to McMurdo or to South Pole, you'll fly through New Zealand. That's pretty much how all the U.S. researchers go down. And so if we kind of get a little bit closer here, we typically fly into Punta Arenas, Chile, and then get on a really large research vessel and go out the Straits of Magellan, actually to the Antarctic, and then down, to the Atlantic, and then down into the Antarctic, all the way to Palmer Station. And so this is about 1,000 miles across. And it'll take us maybe 5 or 6 days, depending on weather, to get down into Antarctica. So this is the research vessel, the Lawrence M. Gould. It's about 250 feet. It's incredibly ugly. It's orange, but it does the job. And we also do research cruises. We also do research cruises on the ship, so it's not only just transport back and forth from Antarctica to South America, it's also a research vessel. This is just a photo taking off from South America through the Straits of Magellan. And as we kind of get into the Drake Passage, I know this is kind of not a great photo, but the birding is really amazing. You know, we'll see things like light-mantled sooty albatross. These are Cape petrels, and these birds really like to follow the ship quite a bit. And they often kind of flock up in these big groups. This is a southern fulmar here. You'll see these guys together quite a bit. And then also you'll see other kinds of albatross. This guy here is a black-browed albatross, and that's another light-mantled sooty albatross. But you know, once we get into Antarctica, really the defining characteristic is that you start to see sea ice, and it's very, very cold. So this is kind of what the landscape looks like. It's very, very barren, really. It's mostly just ice cap and rock and ocean. But, you know, when we talk about what is environmental change in the Southern Ocean, what we're mostly talking about is variation in sea ice, which is what we see here. And so sea ice actually has a whole ecosystem that's associated with it. So in the summertime, we actually get phytoplankton blooms associated with sea ice. I don't know if you can tell, but this is a really brown, greeny color here in the water with the ice. And so we get certain other types of species that are associated with these types of phytoplankton blooms that are associated with the ice. So certain krill species, silverfish, which are kind of like herring, and then also predators that are associated with these kinds of habitats. So Adelie penguins and emperor penguins are the two only sea ice obligate species of penguin in Antarctica. And this is a Waddell seal here. And so what we're really interested in doing is trying to understand these relationships between changing sea ice and then the trophic relationships between the different food web components and how it really drives the populations of these predators. One other thing I guess I'll touch on at the end is this idea that with a warming environment, at least along the western Antarctic Peninsula, is that we also get increased snowfall at penguin rookeries. So this is just an island near Palmer Station where we work. And one thing that we've noticed is that there tends to be more snowfall. And it really can cause a lot of problems with them when they're breeding because it'll flood nests and then the eggs don't hatch. So I'll talk a little bit more about that later. So in terms of sea ice variability in Antarctica, this is sort of what we know in a nutshell. These are 30 years of integrated data of sea ice around the continent. And so on the Antarctic Peninsula, we've seen actually a 3-month reduction in the sea ice season. Okay? So over 30 years, we're getting 4 less days per year. So you can add it up over 30 years and it ends up being about a 3-month difference. Whereas over in the Ross Sea, the sea ice season is actually growing. So there's a fair bit of variability depending on where in Antarctica you're looking. Some of the declines along the peninsula are as marked as areas of the Arctic. So this is the Chukchi up here, and mostly, there's been sea ice loss in the Arctic. What's interesting along the peninsula is that there's a latitudinal gradient in sea ice variation. So we've got these 3 main study areas that we work at. So the north at Ambers Island, sort of the midway along the peninsula at Avian Island, and then very far south along the peninsula at Charcot Island. And so you can see this graph over here on the right is just showing sea ice cover in percent. So that's this axis here. And then we've got the month of the year on the X and then the year on the Y. And so what you can see is that as we go south, there tends to be more winter sea ice coverage. But also as we go from historical years into like more current years, there tends to be less sea ice forming in each of these 3 regions. And what we think is happening is that sea ice variability is really linked to climate. So one of the climate modes that's really important determinant of sea ice variability is a southern annular mode. And this, when it's in its positive phase, is really associated with a strengthening of the westerly winds around the continent. So as the winds increase, we tend to have warmer winds get picked up from the ocean here and they kind of slam into the Antarctic Peninsula here. But then as the wind comes around the continent, we actually get colder winds forming around the Ross Sea. Also, when ENSO's in its La Nina phase, which it has been more often, we also get more heat transport, which tends to actually kind of run into the peninsula here as well. So these issues of sea ice and climate are really integrated through what is known as the Antarctic Circumpolar Current. And so this is this big body of water.
It'll go away.
Okay. This big body of water here that runs around the continent. And what happens is, this is very cold water, but it's relatively warm compared to the shelf waters, which actually freeze in the wintertime. And so in many areas of the continent, this body of water is kept away from the coast through these gyres, but only along the Antarctic Peninsula does it really intrude right along the continental shelf. And so when we get increasing wind and other types of climate events, what happens is this relatively warm water gets sort of dumped up onto the continental shelf, and it affects the way sea ice forms, particularly in this area of the continent. And so what this graph over here on the right is showing is essentially the bathymetry of the western Antarctic Peninsula. And so as we get more intrusion of this Antarctic circumpolar current water onto the shelf, what we end up seeing is that it gets transported across the shelf in these deep submarine canyons. So this is Palmer Station where we work, and you can see that there's a deep hole right here. This is called the Palmer Deep Canyon. And so this relatively warm water comes across the shelf and then it upwells right here by Amber's Island. And a lot of the penguin colonies along the peninsula are associated with these upwelling features. So what happens with climate variability and increasing water temperatures, it just gets transported across the shelf and then upwells right by these penguin colonies. So we work at Palmer Station here. Avian Island down here is in Marguerite Bay. This is the Marguerite Trough that comes across here. And then we started doing some work down at Charcot Island here and have just sort of recently discovered that there is a deep canyon near this penguin colony as well. So with all of these issues in changing climate and changing sea ice, we've also seen changes in numbers of breeding penguins in this region of Antarctica in particular. So in the north, at Amber's Island, what we've seen are these declines in Adelie penguins, so that's this guy here, up to about 80% of what the historical breeding population was. So they've used to be about 15,000 pairs, and they're now down to under 2,000 breeding pairs. And as I mentioned earlier, Adelie penguins, like emperor penguins, are the only truly sea ice obligate penguin. Antarctic penguin. And so then we also have chinstrap penguins and gentoo penguins. And so gentoo penguins have skyrocketed in numbers, while Adelie penguins have declined, and then chinstrap penguins have increased, but not quite as dramatically. So here in the north, we have these shifting population trends of these three different species, with the chinstrap and gentoo penguin being considered a more, or a lower Antarctic penguin, if not more of a sub-Antarctic penguin. And the picture here on the right just shows you some of the islands that the penguins nest on near Palmer Station. So they're typically ice-free and they have lots of pebbles on them, and that's what the birds use to make their nests. These just show the population trends for the three species since about the mid-70s, when my advisor, Bill Fraser, first started working there. So there used to be about 15,000 Adelie penguins, and really none of the other species. And then you can see it's declined a lot for Adelie penguins, and then increased since about the mid-90s for gentoo penguins. So it's kind of a system that's in transition from what was a polar system into a more sub-Antarctic system. And we can kind of understand these ideas of changes in populations mostly from where the birds are naturally distributed. So for example, Adelie penguins, like emperor penguins, are distributed at really high latitudes around the continent, around the coast of Antarctica. Whereas gentoo penguins, their distribution is more northern, so they really exist at their southern range margin around Palmer, whereas Adelie penguins exist really at their northern range margin in this same area. Further south at Avian Island where we work, I hope you can see this photo because it's actually amazing. There are over 70,000 breeding pairs of Adelie penguins at this colony, and it's the largest Adelie penguin colony on the western Antarctic Peninsula. The other two species, their range doesn't extend this far south, so this is really truly more polar habitat here. And one thing you'll notice about Avian Island is it doesn't get the snow that we get up north at Palmer Station. And this is only about 400 kilometers different, so it's actually not that far apart, but it's really different ecologically. And so I guess I should say, sorry, the Adelies here have been increasing in comparison with declining populations at Amber's Island. And then even further south, at Charcot Island, we think this very small population has actually been stable. And so this is a really fascinating story. We just started working there about 2010. And so there was an early record from the British Antarctic Survey about a penguin colony existing at Charcot Island, and no one else had ever been there. It was just sort of like a one-off observation, and so we got permission on our oceanographic cruise to go down there and to look for this colony. And so we ended up finding the penguin colony. It has about 30 breeding pairs, 60 penguins total. But what was more fascinating about it is, and this came out in this paper here, was we wanted to test this idea that there was this submarine canyon system associated with the breeding penguins at Charcot Island. And so we went there, and this place was actually covered in sea ice. We almost couldn't get in, except that we got close enough, and there was this sort of open water area, which ended up being associated with a marine canyon. And then what we found was that, taking like CTD data, is that it had this signature of what is the Antarctic circumpolar current water, which is relatively warm. So for example, here we are in the north, this is the Palmer Deep Canyon, and if you take data from, these are actually from an autonomous underwater vehicle, so it gets temperature, salinity data, and you can see that right near the penguin colony, the temperature gets really hot, and that this is coming across the shelf, and then we have hot water upwelling. Same thing is true at Avian Island. We get lots of hot water upwelling here closer to the penguin colony. And then we went down and tested this idea with the birds at Charcot Island. And so this was our very first CTD cast there, and sure enough, at depth we have relatively hot water, and then it kind of comes back up closer to the surface. So it seems like Adelie penguins are foraging in these areas associated with upwelling and water that comes from the Antarctic circumpolar current. So just to kind of recap, we've got lots of changes in numbers of breeding penguins across the peninsula and this sort of latitudinal scale that's associated with these differences in sea ice. But what I really wanted to understand was what's the mechanism that links all of this together. And so there's been work by, folks at British Antarctic Survey who do incredible work. And really, this one paper kind of spelled it out for me in saying that what we really need to, in order to understand penguin response to climate variability, is understanding how changes in the marine system affect the demography of the penguins. So whether it's reproductive survival or whether it's the birds moving. So you can imagine this is just kind of a model that I put together that shows you have a species distribution, and when we get changing environmental parameters, it can take it can change one area which used to confer high fitness into an area that's now low fitness. And then if we have changing environmental parameters on the other end of their range, it can go from what was an area of low fitness to an area of high fitness. And this is how we think of range dynamics. And so at Palmer Station, what we typically see is a range contraction, and then for the other species, a range expansion at their other geographic range end. So, what do we know about changes in marine food webs? Well, we know a fair bit from some of the work that the LTR has done. And so this is a paper that actually has been looking at phytoplankton changes over the last 30 years. And what we've seen associated with sea ice change is that in the north, we've had reductions in the production of phytoplankton, but increases further south. And then we also know from some of the predator work we've done with penguins is that in the north, so these are from diet sampling, these are data from diet sampling penguins, especially Adelie penguins. At Amherst Island, historically diets used to be about half Antarctic silverfish, and now they're mostly comprised of Antarctic krill. But as we go further south, the diets in the south are actually more than half still Antarctic silverfish. So we see these diet changes also associated with gradients in sea ice. And then, this has kind of been our model for how we're trying to understand the system, and it's kind of grouped by these different trophic levels. So we've got a time series here showing years from 1970 to 2010, and then this is like chlorophyll here, going from what was high chlorophyll to low chlorophyll, changes in Adelie penguin populations, which is in blue, and it's kind of chopped up between these different trophic levels. But what I wanted to do is try to integrate the whole food web using stable isotopes. And so a lot of my work is focused on looking at carbon and nitrogen stable isotopes, which is something that we can measure in an individual penguin tissue that tells us about sort of the relative trophic level it's been feeding at, or the source of the primary productivity in the system. And so what I ended up doing was working in the summertime when the penguins are breeding, and basically showing up and we monitor study nests. So this is kind of an example of one of the penguin nests. They build these pebbles and then lay 2 eggs in the nest. And then they'll incubate the eggs for about, well, probably at least 30 days, if not more. And then the chicks hatch around December, and then they grow up into January and into February. So we were going around and monitoring these study nests, and then when the adults reached the 1 egg stage, we would capture the adults, weigh and measure them, and then take blood samples from them, and then follow these nests as the chicks grew, and capture the chicks at day 5 and day 15, and as they grew up, and also taking blood samples to look at isotopes and some genetic parameters and physiology about the birds. So I guess I should also say most of this work went on at Palmer Station, so we could work with all 3 species there. But then later on in the season, we would work off the ship, and so this is like later in January, and we can work with the older chicks across the peninsula at those 3 different sites. And so one of the big questions was, are these populations with differing trajectories really linked to different food webs? Because that would implicate, climate and changes in food webs as a potential driver of population change. And so this was kind of one of my big chapters in my thesis. Again, we worked at here in the north and then across these 3 sites. And I guess I should just say that most of my data come from these seasons when it was extremely low sea ice. So 2008 was the lowest sea ice season on record within the 30-year record where we were. And so basically these are what some of the data kind of look like. So these are blood isotope values of the 3 different species at Palmer Station at the 1 egg stage. So these are for adults. And these are the differing sea ice conditions for the winter prior to our sampling. And so what we see is that Adelie penguins, which are in black, they actually foraged at a relatively lower trophic level during the low sea ice year, whereas the opposite was true for gentoo penguins. And then chinstrap penguins were actually totally independent of sea ice conditions. So we know that coming into breeding, they're actually linked to very different food webs at this time of the summer. And I just wanted to note that what's interesting about this is that for both chinstrapped and Adelie penguins, their eggs actually are really pale yellow colored. But for gentoo penguins, their eggs are a very bright orange color, which also suggests that they're being built from different food webs. People have actually looked at carotenoids and what the relative trophic level is, and shown this actually in seabirds in the North Pacific. And so it would make sense that the birds here, Adelies and chinstraps, that are foraging at a relatively higher trophic level would have pale yellow eggs. So that was pretty cool. What we found at Palmer Station in the north is that by the time the chicks were really old, in late January and in February, at least in the last 2 years of the study, the chicks became really similar in their isotope signatures, which would make sense if all the parents are foraging from a very similar source near the breeding colonies. But what was interesting is when we looked at Adelie penguins across the peninsula, so this is Palmer Station in the north, Avian Island sort of on the middle part of the peninsula, and then Charcot Island in the south, you can see that with this gradient of sea ice, we also get this gradient in isotope signatures of the chicks themselves. So it suggests that the parents are feeding these chicks very different things that are driving their isotope value. So to kind of sum up some of this stuff, we saw the parents at the 1 egg stage being linked to really different food webs, and then the chicks from these parents being really similar by the time they're old. But for Adelie chicks, they actually become enriched as we move further south. So there's a lot of food web variation that's associated with these penguins with different population trajectories. And so the answer to the question was, well, are they linked to different food webs? Well, yes, certainly at the early stage of breeding, but not so much for the 3 species around Palmer Station, but yes, that's true for Adelie penguins across the peninsula. And then I wanted to go on and ask, well, does any of this really matter to how well these different species produce their chicks? So we know that if you can raise a chick that's in really good quality, in good nutritional quality, when it goes and fledges from the rookery, it has a higher chance of surviving. And so a lot of these population changes in penguins have been cast in this idea that there are changing food webs, and due to the changes, they don't raise chicks in as good conditions, so they don't survive, and then they don't come back and breed. And that's why the populations are changing. So what I found, I used these same data with isotopes and looked at how well it predicted actually body mass and egg mass of the parents as well as that of the chicks. And so the answer is that variation in foraging really is only a strong predictor of body mass for 5-week-old chicks of Adelie penguins across the peninsula. So these are, in the gray, chicks that were raised from Avian Island in the south, and then these are chicks that were raised in the north at Palmer Station. For all the other stages and species, there really wasn't a strong predictor of foraging within the species of what its body condition would be. So we went on to do some work, some hormonal work with these guys. So we looked at corticosterone. And cort is really a hormone produced by the HPA axis in birds. And it's associated with things like mobilizing energy, but it can have costs, so things like suppressing immunity. And so what I was interested in looking at was whether or not we'd see these sorts of things in the different populations with, say, for example, Adelie penguins at Palmer Station that are declining might have higher cort than the other species. But actually what we found out was that cort does not predict body condition at all in any of these species. So it really suggests that when the penguins are raising their chicks, they're really in good, adequate physiological condition despite all the food web variation we've documented. And that really changes in the population might be due to dispersal more so than effects on performance and whether or not they survive. So the last part of the work really asked questions about dispersal in Adelie penguins only, because we have, we looked at dispersal in Adelie penguins only, and that's mainly because we worked at all these different colonies across the peninsula and could look at the population genetic signature at these different areas, whereas the other species, we only worked with them at their most southern range margin and didn't access some of the other colonies to the north. So I did this work with a lab at USGS in Anchorage, and we used essentially genetic markers, so microsats and mitochondrial markers, to look at really the genetic structure, the spatial genetic structure and gene flow of these different rookeries. And what we ended up finding was that for the most part, there isn't really a lot of genetic structure in Adelie penguins, but we did notice a lot of structure, at least in mitochondrial DNA, in terms of populations that were compared with Charcot Island in the far south. And so mitochondrial DNA is only passed through the maternial lineage. So it's kind of suggestive of this idea of sex-biased dispersal, so that males disperse and females are philopatric, and that's why they would have structure in their mitochondrial markers, which is really super interesting because in these areas where we've had a lot of sea ice change, we actually detect no structure. So what we think we're kind of getting at is this idea that changes in food webs and sea ice are really affecting more the dispersal behavior of the penguins, which is driving their genetic structure, if that makes sense. And then the last analysis we did, we looked at gene flow between at least Amber's Island in the north and Avian Island, and actually we just found that Amber's Island is a sink. So it receives more immigration than it gives out. So it just suggests more individuals are being, I guess, recruited into Amber's Island. But so just to kind of sum up for all this stuff is that we detected a lot of food web variation among species and regions, particularly among adults, and then also the 5-week-old Adelie chicks between the north and the south. But that food web variation really isn't a strong predictor of breeding performance, except when you look at it at the regional scale among Adelie penguins. And then we didn't find any role for stress hormone modulation of body mass, which just means that the penguins are really of fine physiological condition. And that, at least for Adelie penguins, they're mostly a panmictic population, but they may show some structure further south. And that's kind of the end of my work there.
What does panmictic mean?
Panmictic just means an interbreeding population. Yep.
Yeah, question about this, the first one about the adults, and because that was so distinct, the three different,
Yep.
that all three species were so different. Were you taking blood, was that sort of an immediate reflection of what they've been eating, or was that from they go to whole different places before they breed?
So it was from red blood cells,
Mhm.
and so different tissues have different turnover rates, and so we interpret it to be about the 60 days prior to when they were sampled. So it's kind of what they're foraging on very late winter as they're probably coming back to the rookeries.
And how far do they forage from the rookeries prior to coming into breed? I mean,
Well,
do they-
yeah, so the three species winter in different areas.
Oh,
And
okay.
so Adelie penguins winter on the pack ice, and chinstraps are in more open water to the north, and then gentoos winter near their breeding colony or along the coast. So really, you would expect them to be quite different, and that's what we found. But it was cool because then we saw how those isotope signatures change over the breeding season. So eggs are produced from these differing food webs across species, but the chicks at Palmer are produced from very similar food webs there because they're central place foragers, right? So they're going to local areas and just coming back.
So they're all eating the same kinds of fish
Yeah.
or crab or whatever.
Yeah. So I guess I was just gonna, I don't know how much more time we have, but I was gonna just talk a little bit more about the snow story. And so I mentioned, with warming conditions along the Antarctic Peninsula, we typically get more snow falling, and it can really impact the birds early in the season when they're trying to incubate eggs. And so this is something my advisors looked at a bit more. And so these are the different penguin colonies around Palmer Station. And you can see that the declines in at least Adelie penguins, like the rate of decline has been a little bit different between the different colonies. So this one here in purple, this is Litchfield Island, which is here. And there are no Adelie penguins that breed there anymore. There were thousands back in the 70s, and they don't breed there anymore. And what's interesting is that all the areas where the penguins bred on Litchfield are in this southwest position, which tends to accumulate more snow. So it's not to say that the whole story is just related to marine food webs. There's certainly landscape effects that are affecting the penguins as well. And then when these colonies get really small, across these islands, there are predators like skuas that will come and take eggs and chicks. And so when they're not in big groups, they tend to be more susceptible to getting depredated by these guys. So it's sort of a combination of things that really happens that drives, a breeding rookery to just go away. And I guess these are just some of the folks I worked with. And then I also thought I'd just throw in some other photos for fun. This is a giant petrel. And so this was not any of my work, but Bill also works with giant petrels. This is one of the islands near Palmer Station. You can see it's really just fantastic scenery. Some of what we do is put satellite transmitters on these guys, and they will go on foraging trips up to about 800 kilometers. So they'll be gone for like a week when they're trying to provision chicks, and it's amazing how far they go. These are Antarctic terns, these guys, and they're in their breeding plumage. And then sometimes we see Arctic terns, and so you can notice this guy, the black cap is not really that well developed and his bill's sort of black. So they're in their non-breeding plumage in the Antarctic summertime. And then these funny guys are called sheathbills, and they tend to hang out around stations. So I don't know how they ever got to Antarctica really, but they sort of like to hide in, you know, structures and things like that, and they're just sort of look like Antarctic chickens. This is a Wilson storm petrel, and these guys are really small, and they tend to pick little bits of plankton out of the water, and so they sort of look like they're dancing on the edge of the water there when they're foraging. And they breed in sort of rocky areas near the penguin colonies, and so you can hear them chirping from the rocks in the summertime. This is an Antarctic shag. It's like cormorant. And they breed around Palmer Station, and they're pretty cool, just like a regular cormorant. This was a king penguin that actually sort of mistakenly, not to say mistakenly, they don't normally hang out at Palmer Station, which is what you see in the back here. King penguins breed like around South Georgia. But this guy showed up, it was probably a young bird, and he proceeded to molt and turn like really ugly and lose all his feathers. These are a bunch of gentoo penguins. So this is the colony out at Biscuit Point that we work on gentoo penguins. And this is so dark. It's a really nice photo. These are Adelie penguins returning at the end of the day from foraging at Avian Island to feed their chicks. So they typically go out during the day and forage and then come in like late afternoon, and then they're usually with their chicks in the evening. And then this is the beach at Avian Island, and so these would be, you know, the birds, they kind of group up at the edge of the beach, and they all tend to dive into the water at once. This is an Adelie penguin at Avian Island, and you can see it's got a satellite transmitter on its back. So we do a fair bit of transmitter work later on in the breeding season and try to figure out where they go, especially when they're provisioning chicks.
How is that attached?
It's attached with, I can't remember the name of the tape. Tessa tape. Yeah.
To the feathers?
Yeah, to their feathers. And so we use Tessa tape. It kind of just,
So it only stays until they molt?
yeah, so they lose it. And at least for Avian Island, we're only there for about 5 days, and so we leave these tags on and then they fall off when they molt. But for the birds at Palmer, we just take them off because we are there more regularly. So these are some of the seals that are around. We don't do any work with seals other than sort of census them. But this is an Antarctic fur seal. This is an elephant seal, and they get really snotty on their nose. And this is a Weddell seal. And then there are whales around. The LTR does have a group that's working on whales. And then this is us heading north at the end of the season on a totally amazing sunny day. And that's it.
You need volunteers?
Dave.
So there's usually 2 chicks per nest?
Yeah, so these are the Pygoscels penguins. They're the only species in that genus, and they all have 2 chicks.
The other penguins have just one, or is that different?
Emperors have one. Macaroni penguins are interesting because they have what's called an A and a B egg, with the B egg being the one that produces a chick. So they lay 2 eggs, and the A egg's a lot smaller, and so they usually just kick it out. So people have suggested that that species is like in transition from a 2-egg species to a 1-egg species in like evolutionary terms. But yeah, so it kind of varies depending on what genus the birds are in.
That was my question, was the 1-egg stage.
Mm-hmm.
So I heard the 2-egg thing, and then I was like, well, is the one egg stage, can you explain that?
Yeah, like why we sampled them.
Why it's called the one egg stage?
Yeah, so their full clutch is 2 eggs.
Okay.
And basically what happens is the male will show up earlier in the season and defend a territory, and then the female will show up and she'll lay 2 eggs. And then as soon as both eggs are laid, she takes off and goes to forage, and the male takes the first incubation shift. So if we wanted to sample both parents, the best time to do that is after they've laid 1 egg, because they're both still there and she's going to hang out a little bit longer to lay the second egg.
Do they lay just one day apart, or?
They are like 2 to 3 days
Oh.
apart. Yeah. And actually the eggs are pretty big, and they have a really thick shell, probably thicker than what you'd sort of think of a normal bird shell. But I think that's just because they're nesting on rocks, and they sort of need it.
Did you have any interaction with leopard seals?
I didn't personally, but we were talking about this a little bit earlier. You know, the leopard seals will attack penguins. So you'll see what they do is they key on to what beaches the penguins are coming and going from, because it's kind of funny, like, even though it's this whole island and you think they just go from anywhere, they don't. They have like the one beach that they go to and from. So the leopard seals will figure out, you know, where they're going from, and they'll attack them. We've seen, them attack penguins quite a bit. It's pretty brutal. They often shake them, and sometimes you'll see like the skin it's like inside out. So if you see like a penguin skin that's inside out, it's usually because the leopard seal has been shaking it so bad that it pulls the bird out of its skin.
I'll just insert what you and I were talking about. If this group, if there's people here who haven't seen this video of the geographic photographer that gave a TED talk of his interaction with a leopard seal, has anybody, have people not seen that? Google it and watch it tonight, because it's like the most amazing human interacting with a wild, what's considered a dangerous animal story you'll ever see. And I won't spoil it for you. Google, it's a TED talk, right?
Is it a TED talk?
It's Paul
I thought
Nicklin.
it's a radio program, but.
It's Paul Nicklin. He's a geographic photographer. He's an underwater photographer, and he was filming leopard seals. And it's a story of this one giant leopard seal that he filmed. And I've seen it presented as a
It might
TED
be on
talk.
the National Geographic website.
Yeah, just Google leopard seal, geographic, and you'll come up with the video of it pretty quick.
Yeah, it's pretty amazing. He's a diver, and so he was
Yeah.
in the water, and this leopard seal was trying to like feed him a penguin, like show him how to attack penguins, I guess.
Yeah.
But yeah,
I felt sorry for him.
yeah, it was really amazing.
That's a beautiful story. So
We
watch.
actually
That's your
had,
homework.
we were out in the little Zodiac, and we've had penguins porpoise and fly up and come into the Zodiac because there's like a leopard seal out there, so they're just trying to get away from them, and they'll just try to go anywhere they can.
Well, after I got a few years ago in a new Zodiac, and it was interesting because it didn't have the pointy end on the stern, how they all have, it was a blunt. And somebody said that, a big part of their market is Antarctica, and the leopard seals will grab and bite those pointy ends on the Zodiac, so they've actually changed the design.
Oh, really?
Because the leopard seals were aggressive to that pointy end of the Zodiac.
Yeah.
Now with the flat end and on the float, they can't get their mouth around it.
Yeah, Palmer, I think we had older Zodiacs models, but they put these really hard cones on the back and
for the
strap
old
them on
ones,
there
right.
because,
Yeah.
the leopards will come and bite.
Zodiac changed the whole design.
Yeah. Huh.
So the back end don't have the points either?
No, the back end's just all flat now.
So
You had a really nice picture leaving South America and then a real nice picture coming into the sea ice. I often wonder about that part in between,
Oh, yeah.
the Roaring Forties. Were you no pictures from that because you were just out?
Yeah, sorry, I should have, what's actually really cool is on the ship, it's got this like continuous flow water monitor, so you can look at the screen and see like what's going on with the water, and you'll know when you hit the Antarctic convergence because the water temperature just dives, and you can feel it when you go outside. It's just, everything just gets cold. And there's this noticeable place like when you're crossing the Drake, that happens.
Just the seas, involve the winds of the seas, and clear
Yeah.
South America.
But you know, the Antarctic convergence, it happens a fair bit north of where you think it would based on where you start to encounter sea ice. So sea ice tends, at least in the northern part of the peninsula, it tends to just be like very close to land, at least when we went down. Probably not in the winter time, but the convergence is a fair bit north of where you start to see ice.
So when you're putting transmitters on penguins, how are you getting them in the water? Or are you capturing the penguins on land or in the water?
So we, this is the cool thing about working with them, is they come back to their chicks. So all the work is just done on land, and it's really easy. I don't want to say it's easy, but it's relatively easy to study penguins in the summertime. And so that's why we know so much about the breeding biology of these guys during summer. Whereas we hardly know anything about them in the wintertime because they're at sea for the most part. So we would catch birds. Well, the idea is when we put tags out, we want to see where they're foraging in order to assess where they're getting prey to feed their chicks. So it's only reproductive adults that we put the tags out on. And typically, yeah, we try to put them out and then get the tags back. So they'll just come back to their nest. And so we'll know, yeah, where they are.
Hide and catch them?
Well, mostly at the beach, actually. We sort of try to stay away from the colony itself. So there's sort of like these islands, and then the actual penguin colony is just like a small sub-portion of the island. And so to be like not as disturbing to the chicks, we would catch adults at the beach. But for my work, we were catching them at the nest, and so you can kind of catch them both places.
But just with the net, or?
Yeah, we had a big landing net. If they're at their nest, you can almost just walk up to them and go pick them up.
But they bite.
They do bite. I have a lot of scars on my hands, actually.
I know you didn't find any relationship with the court work, but were
Yeah.
you looking at plasma or feather or fecal? Like what kind of scale were you looking at?
So I took all kinds of blood samples, and so we looked at plasma cord.
Okay.
And so we did, as you know, the hard sampling of trying to get it under 3 minutes. And it was hard, actually, to do that, especially for the adults, because you're grabbing both adults, and they're really strong, and then you're blood sampling one and then trying to blood sample the other. So what we ended up doing was we had this big wooden box that we made with like holes in it, and so we put one adult in there, blood sample one, and then switch them out, and then after we weigh and measure everybody and put in the egg, we put the egg back, and then amazingly enough, after you handle them, they just go back to their egg. Like you just put the egg back and then release them, and they just go back to their egg. So.
I haven't done any work with court, but does the stress of capture influence the accuracy of that result?
Yeah, so that's why we like to get blood samples in under 3 minutes. And so there's been a fair bit of work looking at what the increases are due just to handling stress, because it will stress them out. And so the idea is if we want to look at stress related to body condition and foraging, it needs to be baseline court, not stress-induced court. So yeah, it can. It's a hard thing to do, for sure.
Well, thank you.
Yeah, thank you so much.
Just mention one more bird sighting I forgot about. Neil Warnock put on Facebook that he's already seen an Arctic tern on the Anchorage Coastal Trail. So they ought to be out in front of us too, and that's a neat migrant that we get back. It's just been so rough, probably no one's been in their boats the last few weeks.
So I did want to mention, this is our last Audubon meeting for this season, unless somebody wonderful wanders through town and we can get them to talk. But we'll keep you informed via email, and always, don't hesitate to contact us.
The next one will then be in the Cordova Center, where
That's
there won't be so
September.
much light coming in.
Right.
Let's hope.
You can open this one?
Yeah.
Okay, so I hope to see you all tomorrow night at the Science Center's birthday party.
It's Earth Day tomorrow, right?
Yes.